Hey, good morning, everybody. Happy Friday to you. I'm glad to see everybody has survived the second week of classes. Um, so let's go ahead and continue our discussion about different examples of important macromolecule polymers. I wanted to start off just with a kind of an example of relating how chemical structures are very important in terms of understanding things like stability of the molecules. So all biological molecules, um, you know, are kind of chosen by life as part of the evolutionary process because they are what we consider to be metastable. Metastable, means that these molecules will stay together more or less with their arrangement of atoms, um, very stably. They don't just spontaneously break down. Because if molecules just spontaneously fell apart and the bonds just kind of broke by themselves, then, you know, one way of thinking about it is it'd be very unreliable to depend upon these molecules, especially when you're talking about large molecules like polymers, right? Because if things just fell apart all the time by themselves, how could we possibly expect a living cell inside of our body to be able to maintain things? So let's talk about this a little bit more, this idea of things being stable and relating this to bond energy. Okay. Here is an example of glycerol. So glycerol, as we were introduced to in our previous lecture when we looked at lipids, is this small 3-carbon alcohol, right? So it has 3 carbons with 3 hydroxyl groups on it, and the other potential valence interactions of these carbons are taken up by other hydrogens. It's a very neat kind of tidy little molecule, right? So you can draw it out this way, or you can draw it this way. This is kind of a useful way of drawing it because you have the 3 carbons and you can appreciate that the 3 hydroxyl groups have enough space in 3-dimensional space to sort of be happy attached to the same 3-carbon backbone. If you look at the little, plastic model, the little balls and sticks here, the red orbs are the oxygens, right? Each with their little hydrogen on them. So these are the hydroxyl groups. You can get a sense that they have enough space to kind of point away from each other so that the whole molecule is pretty stable. And that's why something like glycerol forms a very nice way of organizing other large molecules, like in this case, 3 fatty acid chains, right? So each one of these little zigzags is a long hydrocarbon chain. Because it has a carboxyl group on it, we realize, oh, this is a fatty acid. Right? This is what we learned about last time when we said that fatty acids are oftentimes an important component for making larger, more complicated lipid molecules. So when you have 3 fatty acid chains that are attached to the glycerol over here, you have a triglyceride, right? And this is body fat. Right? So I think we all know, hey, unfortunately, something like body fat is pretty stable. Right? Oh boy, don't we wish it was easy to kind of break down body fat. Right? But that's why evolutionarily, or something like glycerol is very dependable. It's reliable, it won't fall apart, so it can be used for something long-term like body fat. What would happen though if instead of these 3 hydroxyl groups on the glycerol, what if you replaced these with something, I don't know, kind of bulkier on them? For example, nitrate groups, right? So here, instead of OHs being attached to each of the 3 carbons, now you actually have the oxygen attached as part of a nitrate group, right? Then NO3. Wow, if you look at the structure now, you can really get a sense that these bulkier groups are going to have a harder time being happy coexisting with each other on this tiny little 3-carbon chain that's holding them together. You can get a sense, right, that they'd be sort of jostling for position in 3-dimensional space. So as a result, the bonds linking them to the 3-carbon chain, the 3 black carbons here, right, there's a lot more instability there, right? Because it's much easier to imagine that the electrons that are involved in this right? If given the opportunity, they would love to kind of be involved in a much lower energy reaction if another molecule came along and you gave the nitrate groups an opportunity to kind of break off. And so sure enough, that's why this is nitroglycerol. And I think, nitroglycerol, at least from like the movies and things like that, right, it's highly explosive. And that's because it's highly unstable. You give this a little bit of energy, right? And these bonds then, the electrons will happily break. And if there's oxygen around, floating around, like in the air, right, they will happily re-engage with bonds with those oxygens. And you'd form things like carbon dioxide and water. And this would happen very rapidly because all these nitrate groups would love to fall off. And as they're releasing this energy, this extra energy, as the bonds reform as water and carbon dioxide, you have that extra difference in energy going from high to low, right? That's released as heat. And that's why you have this violent kind of reaction. And it's very explosive. So nitroglycerol is not very practical for different industrial, explosive uses because it's just too unstable. Right? So what if you were to compromise? What if you said, well, glycerol is useless as an explosive agent, right? Because it's just too stable. Nitroglycerol is too unstable. So what if you did something like this, right? What if you had the same 3 nitrate groups attached to carbons, but this time instead of having them stuck on just 3 carbons, you have 6 carbons in a ring, right? Now look, because of this ring structure, the nitrate groups have a little bit more stability, right? They each have their kind of space to be in apart from each other, right? So this is trinitrotoluene, or TNT. So that's why when TNT was invented, right, it was a much more practical, manageable explosive for different types of purposes. Again, because of the chemical structure, even though the types of bonds that we're talking about breaking and reforming here are the same types of bonds. So this is just a little note that TNT is actually not the same thing as dynamite. I think I always thought TNT and dynamite, it's like the same thing. Right? Dynamite is actually nitroglycerol mixed in with different inert, non-reactive components to kind of, mellow it out, if you will. I think originally dynamite was nitroglycerol mixed with different types of sand and things like that to kind of stabilize it. Right. So that's just a little bit of trivia, right? But the important thing here that's not trivial is for you to begin to appreciate this idea that 3-dimensional arrangement of bonds has a big influence on things such as the amount of potential energy that's available there to be released, and whether or not the molecule is stable or less stable. Okay. And as we discuss different biological molecules through the semester, we will see examples of this, right? Why some molecules are actually used for certain functions inside of the cell because they have higher potential energy in them, even though they're made out of the same carbons, hydrogens, nitrogens, phosphate groups as other molecules that we talk about. All right. So let's expand on this idea of 3-dimensional shape of molecules and why it's so important for us to get away from always thinking about molecules just as little line diagrams, carbons with little lines attaching to oxygens and things like that, right? In Bio 275, as molecular cell biologists, we have to think of 3-dimensional shapes, right? Because that's what really matters in our living bodies. So recall that function depends upon the proper shape of the molecule so that it can interact with other 3-dimensional molecules. How do we actually know the 3-dimensional shape of these molecules, right? Because, they're down at the level of atoms, right? How can you actually see these things, right? And even then, if we talk about, the 3-dimensional shape is actually the electron orbitals, how do you see those things, right? Our technology is not at a point where we can actually see something like individual atoms, in with their orbitals making a 3-dimensional space, right? But we're getting there. But certainly, for a long time now, we've been able to determine the shapes of important molecules like proteins. So how have we been doing that? So here's an example. This is hemoglobin, we used hemoglobin as an example before when we were talking about the fact that amino acids that are strung together with peptide bonds to form a peptide chain, that polypeptide chain then will fold up as a complete protein. And the way it folds up is dependent upon things like what different amino acid sequence you're looking at, because each of those amino acids has different R groups sticking off. Some of them are charged, some of them are not charged, some of them are bulky, some of them are not bulky. And as a result, the chain, only has a certain number of different ways it can fold up, especially when you consider that it's surrounded by water, and water is going to be interacting with hydrophilic side chains and pushing away hydrophobic side chains to help influence how something like the hemoglobin folds up into a tertiary protein structure. So how do we know the structure of this? We can represent hemoglobin either as a space-filling model when we think about the electron clouds, or as we've been talking about in this class, sometimes it's more convenient just to try to follow what is the polypeptide backbone, right? If you just thought about for each amino acid, the standard carbon that has a amine group and a carboxyl group and a hydrogen, right? All amino acids, the 20 amino acids have those. It's just the extra R group that varies. But if you were to follow the links from the amine group of one to the carboxyl group of the other that form these peptide bonds, right? You can form a ribbon diagram like this, right? In this ribbon diagram, each one of these different colors is a different hemoglobin polypeptide, right? And they come together to form a quaternary structure, a complex protein made of, in this case, 4 separate hemoglobin proteins coming together. And that's what these ribbons are, showing the peptide background. So simply knowing the amino acid sequence is not enough, right? We would have to predict the overall 3-dimensional shape using some sort of other means. Traditionally, historically, we've used a technique known as X-ray crystallography. So X-ray crystallography, as the name implies, has to do with beaming X-rays through crystals. Now, crystals, I'm not talking about new agey kind of crystals like quartz and stuff like that. I'm talking about if you take biological molecules like proteins, like hemoglobin, that have a very stable 3-dimensional shape, right, you can encourage those to crystallize, meaning you try to remove a lot of the water and the resulting purified hemoglobin molecules will naturally kind of stack up in a repeating pattern. Right? If you then shine an X-ray or X-rays through this repeated pattern, which is a crystal of this protein, you can imagine then, because there's a repeating lattice work, right, a certain geometry of repeating patterns inside the crystal, the X-ray beams will diffract in very uniform kind of ways. Right? If you then are able to capture the way that these X-rays have diffracted, for example, on radioactive sensitive film, like X-ray film, right, you can get a pattern. And if you know how to interpret that pattern, you can actually work backwards and figure out, the molecules inside this crystal most likely had this particular shape in order to result in this kind of shadow pattern, right? Kind of neat. Right? So here's a crystal of some protein. Let's pretend it's hemoglobin, right? So it's made up of many, many, I don't know, billions of or millions of hemoglobin protein molecules that have formed this crystal. You shine X-ray through it, you get a diffraction pattern. And these days, of course, we have sophisticated software that can interpret this pattern and kind of work backwards and figure out what the different types of bonds and shapes would have to be in this crystal. And then you could generate then the predicted 3-dimensional shape of this protein. Now, that's how we do it today with modern software, right? But people have been doing X-ray crystallography since, I don't know, like the early 1940s, certainly well before they had fancy software. So those people, right, I mean, they were obviously limited in the types of shapes they could figure out, but they were really well versed, really well trained in recognizing certain patterns to indicate certain features of three-dimensional shape. And that's what I want to show you an interesting example of today. Okay. So this is the very first image captured through X-ray crystallography of hemoglobin. So this is in the 1950s, right? So from this, the X-ray crystallography, experts were able to tell that this type of diffraction pattern meant that hemoglobin, the molecule, was probably shaped in a certain way. So around that same time, there were other advancements that were happening due to X-ray crystallography. And so this is the story of a very important one. This is a copy of Nature. So Nature is one of the premier, most highly regarded peer-reviewed scientific journals. So Nature's been around for a long time, but this particular issue dates to Saturday, April 25th, 1953. So if you look at the table of contents, you can kind of get a glimpse, it's kind of amusing actually, of what the important scientific sort of cutting edge discoveries were, back in 1953. So here we have, for example, government scientific research going on in Great Britain, okay, prehistoric America and Polynesia. Oh, that's kind of interesting, right? Even back then, they were talking about the relationship between Polynesian cultures across the Pacific and prehistoric America, as they call it, right? Let's see, impact of man on British wildlife. I'm sure that's still an ongoing problem. Let's see, developments in methods of microscopy. This one's kind of interesting, deodar as pencil wood. Okay, right, I don't know what deodar is, but I guess back then maybe it was really important to find out if it worked well for making pencils, those high-tech things, pencils back in 1953. Okay, well, anyhow, tucked all the way down in here is this article, kind of very modestly, very far in this journal, and it's Molecular Structure of Nucleic Acids, right, by these unknown scientists, J.D. Watson and F.H.C. Crick. Okay, so what is this article? If you actually flip to whatever it is, page 753, right, here's the article. Right, so of course you guys all know Watson and Crick, or I mean, you might not know their story, but they were the people who discovered the structure of DNA, and they did so when they were pretty young. I think Watson was still a graduate student, and Crick was a postdoc, right? But they are the first ones to predict the structure of DNA, and they got it right, because many before them actually tried to come up with structures that they thought made sense. So Watson and Crick, they were not the first to propose a structure of DNA, right? But their version of DNA was pretty radical. Here's the article here, Molecular Structure of Nucleic Acids. Right? And you can see the article is actually not that long. I think by the end of this column on the second column over here, I think after this, they just go through like a paragraph of thanking people or whatever. And this is the only illustration, right, in that article. And I think all of you recognize this now as being DNA. But just think, back in 1953, this was the very first illustration of DNA, right? The general public, if you showed them this, they would have no idea what it was. Right? And look how simple the drawing is. There's no fancy measurements or anything. It's just the alpha double helix. They have like a little arrow showing that one strand is going one way, the other strand goes the other way, and it's linked together by these rungs, like the rung of a ladder that's twisted around. All right. And that's it. Okay. I mean, that's pretty amazing if you think about it, right? I often thought, hey, wouldn't it be cool to have a copy, an actual copy of this issue of Nature? But then like on eBay, when they do show up, they go for like $6,000 or something like that, right? So I'll just settle for this PowerPoint photo that I got off the internet. So Watson and Crick, they based their model on X-ray crystallography results. So this is where the story gets really kind of interesting, because they were not the ones to generate the X-ray crystallography data. In fact, they were not even trained on how to actually do the X-ray crystallography, right? They actually benefited from the work of somebody else. And that somebody else is Rosalind Franklin. So Rosalind Franklin was a junior scientist who worked with some big shot researchers whom Watson and Crick, through their boss, kind of knew also. And one day, I think it was Crick, or maybe it was Watson, one of them was visiting the lab where Rosalind Franklin worked. And Rosalind Franklin, she was a real expert on X-ray crystallography. This was her thing, right? She loved to take different things, try to see if she could make crystals out of them, and then she would shine X-rays through them. And she would have this big collection, huge portfolios of different images. And so she crystallized DNA, right? And she did her X-ray crystallography, and she had a whole series of developed film. And one of them, this photo, which is photo number 51, right? When she showed this to Watson and Crick, right, suddenly a light went off in their head. Because even though they didn't know how to generate these X-ray crystallography patterns themselves, right, they had read up enough on these. And when talking with Rosalind Franklin, they came to the conclusion that this sort of hash mark X pattern, right, of X-rays being diffracted through the crystal could only be the result of an alpha helical structure. Okay. Francis and Crick went on to win the Nobel Prize. And I think, unfortunately, for many decades, Rosalind Franklin did not get the credit that she deserved. Now, fortunately, now these days, people, when we look back at history, right, I think we give Rosalind Franklin a lot more credit now, the credit that she rightfully deserves for generating this critical piece of evidence that allowed Watson and Crick to get their model correct. All right, so that leads into our discussion today of nucleic acids as our fourth example of important macromolecule families, and it is our third example of polymers. So nucleic acids, like DNA, are made up of polymerized chains of these building blocks that we call nucleotides. So I think all of you have gone through, at least even in high school biology, but certainly if you've already taken 171, 172, this part would just be review, hopefully, right? So DNA is a polymer chain made up of these different nucleotide units, individual monomers, right? And these monomers consist of different parts that make up the individual monomer. Part of that is a nitrogenous base, so either an adenine, thymine, cytosine, or guanine, right? And that's attached to a sugar group, which is not pictured in this cartoon here, but those in turn are attached to a phosphate group. And it's the phosphate group that's crucial for linking the monomers together to form a polymer chain. Okay. So the 5 nitrogenous bases that make up our nucleic acids in our cells, our DNA and RNA, right, here they are pictured. Here are the structures. Cytosine, thymine, right, and uracil are the pyrimidines. Okay. I think you all know that in DNA, thymine is used. That's the T. Whereas if we're talking about RNA, instead of a T, it uses uracil. And you can really see how similar the uracil is to the thymine, right? The difference mainly is the thymine has a methyl group attached there. The other 2 nitrogenous bases are the purines, so adenine and guanine. Okay. So I need you to be familiar with the structures of these 5 nitrogenous bases, right? I need you to be able to recognize them by sight, because as we start talking about things like DNA sequence and why the different choices of nucleotides in different positions are important for resulting in the expression of different kinds of protein products or gene products from our DNA, it only makes sense if we can appreciate the structure of these. right? And how the structure of these are important for how the message is passed on when you're coding for protein products. Okay. I think an easy way to remember it is, it's only 5 structures and they're very similar. So you just remember key things, right? Like the methyl group on the thymine. If you don't have the methyl group there, it's a uracil, right? And then the cytosine has the amine group at the top. And then the purines, I often remember, if you just remember purag, purines are adenine and guanine. If you just remember that, then the other 3 are automatically pyrimidines, right? So a nucleotide then, in order to come together to form a functional monomer that's used as part of making a nucleic acid, you would take one of these nitrogenous bases, okay, so as pictured here, you would attach it to a 5-carbon sugar. So so these are based on ribose sugars. So ribose sugars are these 5-carbon sugars, right? And if you have a hydroxyl group at the C2 position in the ribose sugar, this is just ribose, right? But if you were to have just a hydrogen in that position, right, this is deoxyribose, right? Because you got rid of the oxygen, right? So now it's deoxyribose. So 1 nitrogenous base, 1 ribose or deoxyribose, and then a phosphate group that, as I said before, is crucial because this is the backbone of the latter. Up at the top here, this just helps introduce some of the different nomenclature that we can use as we talk about something like an individual nitrogenous base. Once you add it to a sugar, right, you start off with a just a nitrogenous base. If you attach it to a sugar, you end up with a nucleoside, right? Because that is different than just the base by itself, right? The base plus the sugar is a nucleoside. And then if you add on that third part, which is the phosphate group, together you have a nucleotide, okay? And as a result, there's oftentimes subtle but kind of confusing technical differences in the names. So the base would be called adenine just by itself. Once you attach it to a ribose or deoxyribose, that combination would become adenosine. Once you add on the phosphate group, it becomes adenosine monophosphate, right? Because you have 1 phosphate group added on. If you had 2 phosphate groups added on, this would be adenosine diphosphate, or if you had 3, adenosine triphosphate, okay? So be familiar with this nomenclature, because the sooner you sort this out in your head, the less confused you will be when you're reading about stuff and it's referring to different things, okay? Here's another way of visualizing it. I oftentimes like to show you guys different ways of viewing certain things that might help you remember it, right? So these are the 3 main components. You have a nitrogenous base, right, a sugar ring, and the phosphate group to form the nucleotide. I want to be very clear about something, and I think this is the first time in this semester that I will introduce this very important concept, and I think it's thought-provoking. You all know there's an A, a T, a C, and a G. You've known that since high school, right? But maybe you've been under the false impression that these are the only 4 or 5, if you include uracil, the only 4 possible nitrogenous bases there are, right? Hopefully you realize, no, why would that be? Because obviously these are pretty simple molecules, right? There are a bazillion, this is what my kids say, they use the words bazillion, almost infinite number of different substitutions of different atoms that you could find, right, in the universe on these rings, right? All of which you would still consider a nitrogenous base because it has the nitrogen groups as part of the central rings. Right? It just so happens, though, that life on Earth, billions of years ago, as life evolved, it turns out you only need to use these 5, right, in order to make life happen. So as a result, biology and evolution is often about efficiency. Okay. So very early on in life, these 5 nitrogenous bases was enough out of the possible infinite number that actually exists. Hey, no doubt about it, right now on the planet, right, we're surrounded by other molecules that look like this, that are nitrogenous bases, but those are just not the ones that living things use. Similarly, right, we already know, like sugars, for example. There are a ton of examples of other sugars. We looked at some of them: glucose, fructose, galactose, things like that, that are also ringed sugars. It's just that the ribose is the one that life, billions of years ago, started using, right? Ribose or deoxyribose. And then you have the phosphate group. So down here, again, is a nice reminder of how the naming works. So if you have an adenine as your nitrogenous base of choice, right, that would be just adenine by itself. But if it's attached to a ribose sugar, then you have adenosine. Right? If you then go ahead and add on your phosphate groups, now you have a nucleotide. From a nitrogenous base to a nucleoside with a sugar group to a nucleotide, because you have a phosphate group attached to it, right? And that can be monophosphate, diphosphate, or triphosphate, right? And adenosine triphosphate is called ATP. Right? And you're like, oh man, why do they call it ATP? Because that gets me confused with the ATP that's supposed to be like the energy molecule of the cell, right? Your cell burns ATPs. You guys know that. Well, don't be confused, because this is ATP, right? This is the same ATP. And you're like, what? But I thought that's like an energy molecule, not like a DNA building molecule, right? But this is another important theme in this class. When Mother Nature, throughout the course of evolution, came across different molecules that are useful, and therefore life counts on these molecules, and these molecules are present in the cell, not surprisingly, after millions of years, billions of years of evolution, right, many of these molecules are used for different functions in the cell. So here you have an example of a nucleotide, right, adenosine monophosphate, that's used as a building block of DNA or RNA. But if you add a couple extra phosphate groups onto it, it no longer works as a building block for nucleic acid. It has another important function in the cell, which we will study later, and that is serving as an energy molecule, ATP. Here again are just some other ways of visualizing the three-dimensional shape. All right, so this with the space filling model is probably the best way of imagining what an individual adenine nucleotide would look like. So as these monomers are strung together to form polymers, you can appreciate here are two examples of such polymers. One's purple, one's peach colored. And you can see the individual nucleotides are strung together. In this case, there's a T, an A, a C, and a G, right? And in this case, there's a U, an A, a C, and a G. Right? The difference being, if you look at the sugar group, this is the ribose ring, because it still has the hydroxyl group on the C2 position. But if you remove the oxygen, in other words, you deoxyribose this, right? This is now deoxyribose. And so this purple one is DNA, the peach one is RNA. Okay. The way that these monomers are connected, again, just like we learned before, monomers in living systems are activated and attached to each other through condensation reactions, right? So some other big molecule is going to actually be attached to these free-floating nucleotides and bring them together and start building this polymer linkage through condensation. So water molecules are released. Right? And so that resulting bond that links the monomers is a phosphodiester bond that involves the phosphate group of one nucleotide linking up with the sugar group of the neighboring nucleotide. Okay. So this is just review, but if you look at something like DNA, double-stranded DNA, I think you're all aware that if you were to look at the individual rungs of the ladder that go across the middle of the DNA molecule, the base pairing between the base on the left and the base on the right here is complementary. Right? A's bind with T's, C's bind with G's. And the way they interact is not through covalent bonds. Right? It is through, again, the little blue dotted lines, which hopefully by now you realize are hydrogen bonds. It's just because of the slight negative, slight positive charges, the polar nature of some of these molecules, you would have these relatively weak interactions. And you can see why the base pairing happens in a certain way, because A's and T's, when they are across from each other in the right position, you can have these hydrogen bonds forming right at these particular hydrogens and oxygens, or hydrogens and nitrogens. On the other hand, C's and G's, when they are in the right position, they form or have the opportunity to interact via 3 hydrogen bonds. That is why the interaction between C's and G's is actually a slightly stronger interaction compared to the interaction between A's and T's. Importantly, this weak hydrophobic interaction, it needs to be weak because part of the way DNA works as an information molecule is that the 2 complementary strands have to be able to separate rather easily. And that's how living things like your cells access the genetic information. That's how the sequence of A's, T's, C's, and G's can be read. Right? Otherwise, if the double-stranded DNA is closed up, that information is not accessible. So importantly, the strand on the left, because it ends up here at the very top of the slide with the phosphate group of this particular adenosine nucleotide, right? This phosphate group is attached to the 5 or the carbon 5 of the sugar, right? So carbon 1 is over here, carbon 2, carbon 3, carbon 4, carbon 5. So we say this end of this strand is the 5' end. The opposite end of the strand ends with just the deoxyribose ring. Right? And this would be the 3' end, the carbon at the 3 position. Right? Another nucleotide might be added onto the end here, right? And that would involve, again, the phosphate group of that next nucleotide interacting with the 3' position of this one here. Okay, so as a result, the strand on the left from up to down flows in a 5' to 3' position. The one on the right is in the opposite direction, 5' to 3', going up the slide. Okay. This slide illustrates the phosphodiester bond. So it's very important that you understand, because again, it's an ester bond. So it's like the 2 R groups, if you want, on either side. And it's happening through this phosphate group. So it's a diester bond. There's 2 of them going on here. One of them involves the carbon 5 position, right? The other bond involves the carbon 3 position of the neighboring nucleotide. Okay. So how much of this do I want you to know? Right? I want you to be familiar with this structure. Right? The truth is you guys kind of have it a little easier because in the normal in-person semester, I would require you to actually be able to draw this, all right, from scratch. Okay. But because we don't really have a great way of you doing that on something like an exam, I'm just settling for the fact that I want you guys to be able to recognize this. So for example, if you were shown different components of this, you could actually say, I know how these fit together, right? I would know, for example, that a nitrogenous base such as adenine would be using this nitrogen on it to interact with the C1 position of the ribose sugar. I would know that the phosphate group of that nucleotide attaches at the 5' position, right? And I would know that the phosphodiester bond that's formed in a condensation reaction to help form this long polymer chain is happening between the C3 and the C5 of these 2 neighboring nucleotides. Okay. So if DNA is a polymer, of A, T, C, and G nucleotides, right, what is the resulting three-dimensional shape, and why is that significant? Okay, so let's look at some of the features, the 3D features of DNA. So DNA is what we call a double alpha helix, right? So it's a helical structure, spiral staircase structure. And if you look at it in a space filling model, I think you can appreciate, right, that it's not a sort of simple spiral pattern. There's actually some different parts of it that make certain segments of it distinct from others. Here you can see, I like this illustration very much because I think this is a nice way of appreciating that these are the phosphate groups, right? So the yellow spheres are the phosphates with their 3 oxygens attached to them, right? So they are forming that backbone, and they'd be attached to a ribose sugar or deoxyribose sugar. So these blue kind of groups here, you know, you could probably count 1, 2, 3, 4, 5. There'd be like 5 carbons in there. That's the sugar ring. And then the light orange part then would represent the nitrogenous base that's attached to them. Right? So again, right, when you look at this, you're reminded, yeah, when you actually get rid of those artificial sticks or lines that we use when we draw things to connect things, you really get a sense of how much more compact the structure is. And importantly, only then can you really appreciate the three-dimensional shape that results. Right? And so you can see that the nitrogenous bases then are positioned so that they can interact complementary across the 2 rungs of the ladder. Okay. So where does this helical structure come from? Right? Why isn't DNA just a flat ladder? Right? It comes from the fact, again, that the individual bonds between the atoms are not really straight lines. Right? They're going to be angles because the atoms that they're linking together want to be in positions that are energetically favorable, stable. I use the word happy because I tend to anthropomorphize everything, right? But it allows us to kind of think about things a little better sometimes. So as a result, the bond angles of these different nucleotides linking together will naturally cause them to form a spiral shape, right? Spiral pattern. So if you were to look at the turning of this alpha helix, one complete turn of this, let's say if you put a little star right here as the start, and then if you were to follow it around, it goes around and back, and then it comes in front again. And once it reaches that same spot again, you put a star there. That's one complete turn, right, of the staircase. There are about 10 and a half base pairs, 10 and a half A's and T's, C's and G base pairs in one complete turn. Okay. In addition, there's some aspect of asymmetry to this. There are regions of the spiral that have a much larger groove on the side, right? There are also parts of the spiral that have a smaller groove, right? So this is what we call the major groove of the DNA and the minor groove of the DNA, right? Why is that important? Again, because in order for the information that's contained in this DNA molecule to be accessed and utilized by your cell, right, as part of it being a gene inside of your cell, there are other molecules that have to come along and be the right shape to bind to these different parts of the DNA so that they can do things like unravel the DNA, right? So that means there are certain things like proteins that are designed to bind to the major groove part. They are literally the right shape to recognize the major group, the right size, if you will. And there are other proteins that bind the minor groove. Okay. Another important aspect of our DNA is the handedness of this. So human DNA is what we call right-handed. Right? What does that mean? That means if you were to imagine that this spiral was in front of you, and if you were to actually grab it with your hand, okay, your right hand, your fingers would naturally curl around in the orientation of this cartoon here, and your thumb would be pointing in a particular direction, in this case, up, right? So if your fingers curl in the direction that the spiral staircase is going, right, your thumb will point in the 5' to 3' direction, right, of the strand that you're following. Okay. We also call this bDNA. Right? So bDNA is right-handed DNA. So there are alternate conformations of DNA, because again, keep in mind that the structure, the three-dimensional structure of DNA, is not only dependent upon the bond angles of the nucleotides being added together because of the phosphodiester bond, the two sides of the ladder, but again, this thing is always surrounded by water, right? So in here, that's really never pictured because it'd just be way too confusing if drawings always had water molecules around them. You can imagine water molecules are interacting with this structure, and the interactions, hydrophilic, hydrophobic, right, help encourage the correct alpha helical structure. Right? So these are 3 examples in this cartoon here of different DNA structures, right? And if I were to ask you, which one of these do you think is the normal genomic DNA structure? Okay. Hopefully, you will have said, well, it's the middle one, right? Because the one on the left, which we call aDNA, has lost the distinction between major and minor grooves, right? It's too much symmetry going up, right? The one on the left, zDNA, looks like this jumbled mess. Right? Keep in mind, it's still DNA. So all of the components that are in there, the nucleotides using their phosphate groups to attach together to make a backbone, and then the bases interacting across, those are still intact. Again, it's just because of things like maybe the lack or the absence of water around it, or other features, such as if this is a big, long DNA strand that's actually itself being folded up or manipulated, there might be parts of that strand that are undergoing tension, physical tension, right, that can cause the shape to temporarily get a bit messed up. Right? And so this zDNA is actually left-handed. So if you were to try to figure out how to put your hand around this, you would actually see that your hand goes much easier around the left side, because I know it's kind of hard to tell, the staircase is actually going that way, sort of clockwise, if you will, going up, right? And your thumb would point up. The aDNA, on the other hand, still is right-handed. Right? So bDNA is by far the most common form that we see in our genome, but these other forms do exist, sort of dynamically, temporarily, in different parts of our genome. Again, because our genome is constantly being acted upon by different molecules that want to unravel the DNA, read stuff from it, etc. All right, so the aDNA and the zDNA can be found in short stretches, right? And it may be that these alternative localized regions of your DNA that are in these funny kind of shapes might actually be a way of your cell to control whether the genetic information that's contained there is accessible or not at that moment for your cell to make use of. Okay, so let me go ahead and stop the recording there today.